nicely to our next speaker, Dr. Kerstin Ludwig from Bonn. She's a group leader at the Institute of Human Genetics at the University of Bonn, and she's one of those geneticists who, during the pandemic, changed her work completely. She's an expert in craniofacial genetics, but she's also a member of the West German Genome Center and a member of the German COVID Omics Initiative, of course linked very much to all the international initiatives already discussed by Kenneth. And she's really moved from, let's say, doing her routine research to developing novel genetic tests for diagnostics and really helping out during the pandemic. And we're very excited to hear your talk, Dr. Ludwig. The floor is yours.
Thanks so much. Welcome to everyone who made it that early to this session. I really appreciate you coming here, and I'm very delighted to speak after Kenny, who presented such a fantastic work on host genetics. So I would like to take you with me on a journey that started 2 years ago and which brought me in contact with things as a human geneticist which I haven't thought of before. I have a short disclaimer. I'm a co-founder of a company last summer that basically now tries to put the results that we generated as part of that study into a true applicative tool and hopefully being applied for testing in other countries also. So let's go back to 2020. As Joris mentioned, I'm a human geneticist, and I work on craniofacial genetics, which has nothing to do with critical care or intensive infectious diseases so far. So as part of that work, I study orofacial clefting, which is a craniofacial defect very, very prominently or frequently observed. And we did GWAS studies. We also did sequencing studies identifying rare mutations in individual patients. So I'm a classical complex trait geneticist. I also used that, or pretty early in the pandemic, I joined some of the studies that Kenneth mentioned, and where we used these tools on complex traits genetics to study host genetics in COVID-19. But the center of my work is, of course, technology. So it's array-based genotyping, it's GWAS, and it's also NGS. So we all here in the room are familiar with the capacity of NGS in terms of delivering huge amounts of data. So when SARS-CoV-2 hit the world, it became clear pretty early on that this will become a major public health emergency. It was first noticed on the intensive care units worldwide, where people really got into the treatment and overwhelmed all the intensive care units. And the problem at that time was that we had so many asymptomatic people that spread the infection without knowing it and actually without becoming ill. So here you see the test capacities for SARS-CoV-2 infections in early 2020. Germany has a population of 18 million people, and we were only able to test 50,000 individuals in a week. So this got more and more over the pandemic. We already had 150,000 tests available in July, and in the highest part of the pandemic, we got to around 1.5 million. What were the reasons for this lack of testing? It was basically a lack of consumables, a huge supply chain issue. I don't know whether some of you have been involved in research during that time. It was really hard to get pipette tips. Even gloves were really, really rare on the market. So there was this lack in consumables, in kits, in reagents, but also in instruments and personnel. So again, I think some of the people here in the audience might have been involved in relocation of testing and qPCR machines to virology departments to help them out. And also trained staff was really, we had a shortage in that. So this resulted in a certain way how we, at the beginning of the pandemic, tried to deal with detecting infected people. So that was a contact tracing approach. So if you had an outbreak, only like one person might have been symptomatic, but you would have other asymptomatic carriers of the SARS-CoV-2 infection, which would of course be able to spread that in their organization or in the school or in their workplace, wherever they were in contact. So the health authorities, what did they do? And at least in Germany, and I'm pretty sure that was the same in other regions as well, they basically tracked down the contact person of the symptomatic person. They might have gotten some tests, they might have not, depending on the capacities, and they actually were put in quarantine. But in that approach, you would actually miss people, and actually those that were outside of these contact circles, and which would mean that you would have a high chance to have corona spread into that organization. What we and others hypothesized is that it might make much more sense to do unbiased screenings in such a situation. So instead of really pinning down everyone by contact and symptomatic status, you could just take all of them, test them at the same time, and do like a very sophisticated quarantine and tracing analysis with a chance to eradicate and contain SARS-CoV-2 infections in these organizations or workplaces pretty early on. Of course, nothing is perfect, so again, here we would also see some infections that get undetected, but if you repeat that procedure a couple of times, then you might get clearance of SARS-CoV-2. And this was not only an idea, that was also supported by epidemiological data. Here is a study from Sten Dinnerson group. He actually modeled what you would need to contain a huge outbreak in a large population. And he found out that if you would test every 9 days or every 11 days or even every 13 days, you would eventually stop an outbreak situation. So here we come, with that idea and the capacities of sequencing in mind, we thought that sequencing devices, and I brought them from Illumina here, would actually be a really good tool to increase the testing capacities. And the nice thing about the Illumina machines is actually that they have very different capacities here. So you have tiny or small devices, but from the mini-seq or the MiSeq, you have middle ones with the NextSeq, and you have really, really high capacity devices with the NovaSeq. So that was the stage where we thought, okay, we have a tool to read out SARS-CoV-2 infections, but how do we get the infection into the system? So how can we actually develop molecules also at the same capacity to be detected on the sequencing devices? What do we need to do? And many of you, of course, know how sequencing works. I would just to remind it for those that don't. In order to be able to use sequencing, you have to generate a molecule that has say sequences called P7 and P5 on their end. Why is that? Because the flow cell that you put into a sequencing machine have these complementary sequences, and you need to have molecules that bind to these sequences in order to get amplified and produce a result. So we need to have something that detects SARS-CoV-2 and adds P5 and P7 primers at their end. The second good thing about sequencing is that it reads out individual bases. That's why it's called sequencing. And you can actually use that information once you have it to first detect SARS-CoV-2, of course, but also use that information to discriminate between different samples or between different people that you analyze. And it is something many of you routinely do when you do multiplexing of exomes, multiplexing of gene panels, or other types of analysis where you combine many different sample types, and you want to basically, after the readout, want to still discriminate between the individual samples. So that is the theory of how we want to get that, but we need to have now a library preparation protocol. And actually a library preparation protocol that is easy, fast, cheap, and can be done in for the number of tests that you can run on the sequencing machines. And here we turned to a method that has been actually invented 20 years ago. It's nothing new. It's probably new to many of us because we have never used it in practice, because in many regions where we live, qPCR is the standard nucleic acid amplification technique. But there is another one that is called loop-mediated isothermal amplification, or short LAMP. It was developed in 20 years ago, in 2000, in Japan, in the company. And it is the same, or it follows this principle of qPCR with the huge advantage that it performs that isothermally. So you don't need a cycling device, you can just use a water bath. For those of you who don't know LAMP, I just briefly introduce it to you. It's a bit more complicated from the way it works, but it also requires primer sets, but instead of 2, as you do require them for qPCR, it requires 6. And that's actually a challenge because you need to design those 6 primer sets. Here you have a DNA or an RNA molecule, and there are 6 primer sites in there, and then you add different primers, and the primers actually have complementary sequences. And the enzyme you use is a BST polymerase that has strand displacement properties. So these 2 characteristics of LAMP are actually very helpful because first of all, there's some strands that form like an extension reaction of qPCR, and then you have another primer from the outer side that actually then displaces that strand and generates a new one. And because these 2 sequences here, which are in purple, are actually complementary, to another sequence on the DNA, you will have these these loops. And these loops are then anchors for round and round of amplification, which in the end yield something like that. Super complex structures that look very weird, but they are, if you put that on a gel and read it out, it would actually look like this. So like a huge smear of different fragments of different sizes. And if you want to have then a specific one, you need to have a PCR done to amplify that out. What are the other characteristics of a lamp that are helpful? It's fast and sensitive. Sensitive means it just can get, it works already with 10, or between 10 and 100 copies. And it's fast because you put it in a water bath, and already after like 30 minutes, you get something that you can measure. It's also very specific because it has this huge number of primers compared to qPCR, and you really need all of them to get a product. So if there's already one that is not working, you will actually not get a product for LAMP. And it has already been used as point of care characteristic, or as point of care tests for some infectious diseases in developing countries, because you can, similar to qPCR, you can just measure it with a fluorescent readout. But that is not scalable, because you would then just really have to look at each individual sample after like 20 minutes or so and monitor the color change. So it's not really, the fluorescent readout is not really practical for a high-scale test. And we want to use sequencing, of course. So summarizing what we know about LAMP is that it works for DNA. It also works for RNA, but then you would have to add a reverse transcriptase. It's isothermal, so you can use a water bath, which is really important when you think about the instrument shortages. And it's very importantly, it's also applicable in unpurified samples. So in order to do a qPCR for COVID testing, you need to do an extraction step, and you can actually not, or you don't need that when you do LAMP. So we wanted to bring these two things together, the LAMP amplification and the sequencing. And here I teamed up with Jonathan Schmidt-Borg, who at that time was actually a postdoc in Aviv Regev's lab at the Broad Institute, but had been appointed as associate professor at the University of Bonn. And when Donald Trump closed the borders, he kind of feared that he would not make it back to Germany. So he packed his stuff right away and came back. And so we met at the University of Bonn. And we brought our two ideas together. And Jonathan had actually already conceptualized a way to introduce barcodes into the LAMP reaction. So the barcode is a 10 nucleotide sequence, which we add into one of these complex primer structures. And it has to fulfill some certain criteria. There's certain GC content. We couldn't deal with homopolymers longer than 4 nucleotides. And of course, we needed to have Levenshtein added distances of at least 3, which means that if an error in the sequencing occurs, there's no risk of misattribution of samples. Again, this is just a picture of how that would look like if all this complex LAMP procedure continues to get produced. And in the end, we would have this kind of structure, or one of these structures that you get with LAMP would look like this. And here we can actually introduce another level of barcodes, because we still have to do one PCR to introduce the P5 and P7 primers that we need. And these are the green dots here. And this is the final product we get. We have P7 and P5 on the ends. We have PCR barcodes, and then we have a LAMP barcode in between. And then we have these two boxes here, the purple and the turquoise ones. And this is the two genome sequence from the virus. So here's just an example of one of these sequences, how it would look like. The barcode in yellow, the viral genome sequences left and right, and then all the others is primer sequence. So now we have to answer some very basic questions, because if we want to do that, we need to know that first of all, the introduction of the barcode does not hamper the LAMP reaction. So here's the standard picture of a LAMP product without a barcode, and that is the same picture with some of our barcodes introduced, and we did not see a substantial difference, at least by eye. Still, there might be the chance that the sensitivity gets lower if you introduce the barcodes. We can't see that by the qPCR alone. So we thought about ways to maximize sensitivity, and again, we used we integrated knowledge that has been around for quite a while, and that is actually that locked nucleic acids, so a certain type of modification to primers might actually increase specificity and sensitivity of assays. So this is here, we did 24 replicates at first and had 8 RNA molecules as input, but no LNA modifications on the primers, and we got 7 out of 24 positive replicates. If we repeat that experiment with LNA modification just on the primers, we already increased the sensitivity about twice. We then did a titration experiment, in order to find out which limit of detection we actually have using LNA modified primers, and this was about a limit of detection of about 18 molecules, which is similar to qPCR, at least in the settings that we have at the University of Bonn. A next concern would be different viral loads. So people have either high viral loads, or if they have, in the middle of their infection, they might have very low viral loads if they are early in their infection, and that's actually what we want to get. We really want to get out those that are early infected. So we need to make sure that the read representation in NGS is not driven by the viral load in the sample, because we cannot measure each sample individually. So we did, again, a titration experiment, and we observed that actually there's only a very minor effect of the load or the input molecules into the readout, and actually if we increase from 10,000 to 1 million, we don't actually see any increase or any lag in difference in the read anymore. So it seems that the LAMP reaction, as we conceptualized it, might be able to encompass the different viral loads in just one experiment. And the final consideration now we have the idea, we have the barcodes, we have to design many of them because we want to be scalable. But do we have to consider some things about the barcodes, or can we just take every barcode available? So we designed 480 of them. We performed LAMP with all of them. And then we found out that a couple, like about 10 or so, did not really yield any results. We were pretty surprised by that, and we looked into their sequences. And what we found is that they all share at their 3' end a GTCC motif, which, when we go back to our on our LAMP primer construct, is actually complementary to the Fib primer sequence. So it seems that there are some intramolecular structures at play that would prevent those barcodes to really engage in LAMP, and we excluded all of those from any barcode design that we have. Final question, molecular specificity. So are we really sure that we only capture SARS-CoV-2 and not any of the other viruses or bacteria that are in the throat? And here we tested different viruses, and we could not find any evidence for amplification except for SARS-CoV-2. Now, this was all on the bench and in the lab, and it's very nice if you do it in the lab, but you really have to go out there and do it on real samples. And I don't know how many of you have been involved in such studies, but it is really difficult to get contact to people that are positive when they are in quarantine. So we teamed up with Ricarda Schmidhausen, who led the public health and hygiene department at the University of Bonn, and who really traveled across Bonn and actually even further to get contact to the people that got infected, and she went into their households and got samples. And we had 57 of those samples, and we took paired swabs. That means these people had to provide 2 swabs, and one we did use for qPCR, and one we did use for LAMP-seq. For the qPCR, we did a standard protocol. We rehydrated the swab into 500 microliter to 1 milliliter PBS, then extracted the RNA in an extraction method, eluted it back in 45 microliter, put it in our qPCR machine, and got positive or negative results. And in the case of positive results, we of course also got a CT value. For LAMP-seq, we introduced the swab into just lysis buffer, took this exact 8.3 microliter of this lysis buffer into our LAMP reaction, incubated it in a water bath, and then pooled it and sequenced it. I now show you the results of this experiment, but I just like to introduce the way I will present the results on this slide. So for each individual, we did 4 replicates for the N gene, which is where our LAMP-seq primers are located. And the 4 replicates will either be white if they are negative, or they will be red if they are positive. And then we also introduced a LAMP-seq amplicon for actine in order to control for the presence of biological material. That's here, and we only have one replicate for that. So that is the result. Many of the samples we tested on the right side have 4 out of 4 red amplicons or red replicates. Then we have a couple of mixed results in the middle, and then we have many negative ones. We also have 2 here on the left side, which are just water samples. So we would expect those to be completely negative. All other samples had at least an actine beta positive result, which means we really detected or analyzed biological material. And that is the results of the qPCR here below. All of those that are negative here are actually also negative in qPCR. And then on the right-hand side, most of the red ones have CT values sorted by their CT range. So the lowest CT value, which means the high-risk viral load, was about 20. And then we also had very, very low viral loads here on the left-hand side. And as you can see, up to a CT value of 33, we catched everyone with 4 replicates. Even up to 36, we catched all of them because a positive sample was deemed from 2 positive replicates onwards. But then we failed to detect 3 that had really, really low viral loads. So that was encouraging because it basically showed that we are almost there where qPCR is. And then we talked to people and they were all like, that's very nice, you did it with 57 samples, but how do you really want to scale that? And that's a very fair question, and so we conceptualized an end-to-end pipeline, which included software for tracing the samples, and also a way to really get into this high throughput format very early in the step. So our idea was, we take the swab, we put it directly in a 96 well plate, and the best would be to really do it directly at the place where the swab is taken. We just sterilize it. So when the plate is full, you sterilize the entire plate for 15 minutes at 95 degrees. Then you do 1 pipetting step. We also, as you remember, we also want to save on consumables and pipette tips. So we only just like 1 pipetting step where we use one of these, where we transfer the samples from 96 well to 384. And then we pool that. And pooling always sounds really, really nasty in terms of lab work, but actually we just when the LAMP rect is completed, all of the samples have barcodes. So what we can just do turning around the plate, putting it for 5 seconds in the centrifuge and spinning it down. Because then you collect all the samples in 1 lid, but as they are barcoded, they all have their own identity. So then you can basically put it in a PCR. Plate have to run a PCR, and then you do a sequencing. So now I'm back with the PCR. We have heard that PCR is really the limiting step, but it's not if you remember that every 384 well plate goes into 1 well of the 96 well PCR plate. So actually the plate that comes from test center 1 goes into PCR 1, the plate from test center 2 goes into PCR plate 2, and the 3 goes into 3. And that makes it scalable because in the end you will run all of them on 1 sequencing device. How is that scalable? You can just do some calculations, and I did them for you here. If you have 96 LAMP barcode primers and a 96 well PCR, or 96 PCR barcode primers, which is something that is readily available, you can already run 9,000 samples. If you increase the number to 384 for one of those, you are already at 36,000 samples. And if you use 384 LAMP barcodes and 384 PCR barcodes, that actually sums up to a possible performance of 140,000. And you can even actually accommodate these 140,000 on a MySeq because you don't need so many reads and you don't expect everyone to be positive. So that was, so far we have done everything with commercially available reagents, but these are not as cost efficient as we wanted them to be. So we, again, put our brains together and thought, okay, where can we optimize costs and logistics? So we replaced the commercial lysis buffer by an open source lysis buffer, which we developed in-house. We found out that another type of BST polymerase would actually be working similarly and is out of patent, so you can actually use it for free, which we call BST Idaho. Then the BST Idaho has an additional really nice characteristics, which we found out is it does not need a reverse transcriptase. It has reverse transcriptase activity by itself. So we could actually remove the reverse transcriptase. And then we did some other more minor things. We corrected the concentration, we reduced concentration of primers of dNTPs and also of the reaction volume, so to make it really, really efficient. Of course, if you optimize these things, this might come at certain costs. And in our case, we had to trade off between reduced costs and sensitivity. So the in-house and actually open source protocol that is available to all of you, has about has an LG of 39 molecules, which is a bit, of course, which is 1 degree or 1 CT value worse than the 18 we had before. But it's still sufficient actually to get everyone that has a CT of lower than 33. And that is in the clinical trial we did with leftover samples from a diagnostics lab. We had about 700 samples there. And all of them have been analyzed with qPCR and LAMP-seq. So CT of less than 33, we got all of them in the range of CT values between 33 and 36. We got 15 out of 16, we missed 1. And then we got like stochastic results. It be higher than 36, but many of qPCR also would get stochastic results in this range. So that was pretty, that's pretty nice. And here's just a picture of how we think, or what we conceptualize in terms of software support. So for testing, everyone had to create a QR code for himself. We provide a tool for that, where people can just register and they get this qPCR code, the QR code. This is the software that is available to the people that handle the swaps at the testing site. So it basically shows you it shows you the position where you are when you transfer the swap into the lysis plate. We have control positions here in H11 and H12. And that is actually the way the lysis plate looks when it's used. So you have this lysis plate that is a deep well plate of 96 positions, and then we have developed these plugs. So the lysis plate is sealed with a heat foil, and then just one by one you pierce through the heat foil, you introduce the swap, and then you close the position with a silicon plug just to make sure that you don't contaminate positions with the others. And then you transport it back in the lab, and from there on everything is automatable because it's already in a 96 well format. We applied LAMP-seq in several pilot studies. We were pretty happy and lucky that we got support from the University Hospital of Bonn, who actually did a pilot study with us testing UKB employees. We did about 25,000 tests in about 4 weeks, which was back in January 2021 when there were no rapid antigen tests available. So that was a really nice thing to give the healthcare workers in our university hospital a good feeling in terms of infection and safe working place. We also tested medical students. We went into a high school of Bonn. We did a school testing where we actually once a week used their gymnasium, the sports arena, to actually test 800 students in 4 hours. And we also went into a setting that was really interesting, which is the meat industry, where they actually there's such a high time pressure to get tests as fast as possible. And here we tested 1,800 employees in 2 hours and to return them the individual results back in the late evening of that day. More interesting, very nice experience was actually also testing an orchestra every day prior to their concerts in Cologne. Just as a short side note at the end, as we use sequencing, LAMP-seq also offers the potential to detect or to follow up on variants. When the first variants came out, we were kind of surprised that we can actually design also amplicons that can discriminate between different of these variants. And that is a picture taken from NextStrain, where you actually see diversity pictures. So that's the SARS-CoV-2 genome, and here at the end, the bars actually illustrate positions in the genome where the strains are really diverse. And you see in the N gene, and if you remember, that's the region where we are with LAMP-seq, there is a huge peak here, and I zoom in, and that's the N gene closer, and this region here actually harbors a lot of variability between the different strains. Our initial amplicon that we used for all the experiments was actually located a bit different, but then we replaced that. We designed new primers, we did all of the work again, and found out that we can actually discriminate between alpha, beta, and delta strains just because of these 10 base pair sequence we read out from the viral genome, because amino acids 203, 204, and 205 actually share different sequences across different strains. So we were able to basically track delta and when it came and distinguish it from alpha, which was available in Germany at the time. And when Omicron came, we were also able to distinguish Omicron from delta, because Omicron is based on the backbone of alpha and shares the sequence here. Unfortunately, now we are kind of stuck because we are not able to discriminate BA4 and BA5 from BA2 because they are not different in that particular area, so we would need to develop new amplicons to track that. And here's also a limitation of all these approaches. The moment you have designed your primers, you probably have the new variant and your old one is not usable anymore. We did that, we just confirmed that this works by comparing our results with full genome sequence data from the same individuals. And to make a long story short, it's 100% concordant. So if we predicted wild type, it has all been the wild type in full genome sequencing and similar to all the other virus strains. So as a summary, we developed LAMP-seq as a testing device for large population groups. It's really it becomes efficient in the way when you start to really test thousands of people. We reached that by introducing a sample-specific barcode very, very early in the process, which enables pooling of the samples in the downstream applications. And we developed an open source protocol. All of you can read that in the paper. And the good thing is that everything works isothermally, so you can just put the things in a water bath, and you don't need to have the thermocyclers. The sensitivity is close to qPCR, although not completely, but very significantly it's higher than the rapid antigen tests. So while we conceptualize the LAMP-seq for SARS-CoV-2, of course, we think that the infrastructure, once it is established, can actually rapidly counter future waves of either a coronavirus, but also others, and can then help to manage pandemic outbreaks. With this, I would like to thank the people. It's not as exhaustive list as Kenneth presented in his talk, but it's very valuable people. It was a very interdisciplinary project that brought together people from the molecular biology, from human genetics, and also from the hygiene and public health sector. And I'm particularly grateful to Ricarda and Jonathan, with whom that would not have been possible. Thanks so much for your attention.
Thank you very much, Dr. Ludwig. Very interesting how you went from the meat industry to the Bonner Orchestra, showing that when it comes to disease, we're all alike. Please, if you have questions, go to the microphone. Perhaps if I could start with a very brief question. I was just wondering whether the sensitivity is low enough that you could also do things like wastewater surveillance and these kind of things.
I think so. We have not tried.
Yeah, you now scaled up for numbers, but could you also, let's say, go for very sensitive? Yeah.
Probably not with that protocol. I think here that's really designed for the high numbers.
Yeah.
And it would probably not make sense for wastewater. Mm-hmm. And I'm not so sure about the interfering substances in wastewater. Yeah. We have not tested that.
No. Okay, interesting. Question?
Yeah, Gerold Brenter, Vienna. Here in Vienna, we have a large setup with gargling. Yeah. Would your approach also work with gargling solution? Do you have any experience?
So we don't have any experience, but we have tested that with some not directly gargling material, but with some surrogate samples of that, and it worked. We have not tested that in an extensive way, but I don't see a reason why it shouldn't, to be honest.
Thank you, Kerstin. Incredible talk. And first of all, I would like to thank you for stepping up as a genomicist in time of crisis while most of us were hiding at home with their kids. Great job. Thank you. My question is, as a human geneticist, you came from a very different field and showed what you can do. Very impressive. Now the question is, you personally, will you go back to human genetics? And then also all of us as a field, will we now always have a session in the human genetics meeting where we talk about viral genome sequencing? Or will we all go back to zero and do what we've always done?
Yeah, so great question. I think it was an experience that I would have not thought about 3 years ago. I think what we should be aware of, all of us, is that we have enormous expertise in technologies, which many other fields don't. And we can bring this in situations of crisis and maybe even in non-crisis situations. Do we need a technology session or every meeting? We should. We should have a technology session, because that, I think, is really, the sequencing as itself is going into so many directions, and I think there's so many great people doing great stuff with sequencing outside of human genetics. I don't think we will have virus sequence necessarily as a topic, but as Kenneth also already mentioned, all these infectious diseases, they have been largely under-investigated at all. Host genetics in sepsis, for example, I was really surprised that we do not yet have any significant findings there. So I think infectious genetics will be one spot, and technology in terms of sequencing in different areas.
Okay, great. Yeah.
No?
No.
Yeah. Is there?
Thank you very much, Catherine. So I'm interested to hear, do you actually want to stick to COVID-19, or do you actually currently plan to also extend to other viral pathogens and to differentiate actually the underlying reason in patients?
Yeah, so the next idea is actually to develop that further into more a differential diagnostic approach, so that you can, for example, as soon as someone gets with a respiratory infection, you can actually see whether it's influenza or RSV or COVID. So that's the prominent direction. But of course, you can think about other applications. Agriculture is an amazing field where you want to actually test large samples of cows in order to detect early infections of agricultural diseases. So that's the way to go.
All right. There is no questions from our online participants, so thank you all very much. This was a really important session. I want to thank both speakers for their work and their contribution to healthcare. Thank you very much, Dr. Bailey and Dr. Ludwig.